Energy, 56. Hayakawa et al. In 1991, demonstrated important changes in the mitochondrial DNA in the liver of mice after the administration of AST. The final sentence of this paper reads, "However, for AIDS patients it is urgently necessary to develop a remedy substituting this toxic substance AST." 57. These results were confirmed by histochemical methods in the same year by Cherian and Gherardi. 58. The toxicity of nucleoside analogues in the treatment of viral diseases was thoroughly dealt with in the following years, and it was proven that the toxic effect causes multi-organic impairments in heart muscle, brain and kidney, as well as in liver and pancreas. 59. Further, it was shown that successor drugs of AST such as DDL and DDC cause the same mitochondrial impairment. 60. Since 1991 it would have been mandatory that not only the pharmaceutical industry, but also the registration authorities seriously consider these impairments caused by long-term administration of nucleoside analogues and provide proof of the incoherence of AIDS patients' death with this drug treatment. In general, this obligation has been avoided, and now they face upcoming connected liability questions. HIV protease inhibitors. A new therapeutic principle in the prevention and treatment of AIDS. According to the HIV model, long precursor molecules of proteins along the multiplication process have to be cut at certain interfaces in order to create functional HIV proteins upon which, ultimately, new high viruses form. Synthetically produced short protein molecules, reproduced after the interface, to be cut from the precursor protein, but which cannot be cut should, according to the model again, inhibit the natural activity of the HIV protease and thus prevent the formation of new high viruses. As a matter of fact, the HIV protease has not been isolated, but has been reconstructed by genetic engineering, upon which it was observed that this enzyme is very similar to the human digestive enzyme, pepsin of the class of the aspartate proteases. The problem of the model is that the one and the very same HIV protease would have to be cut at completely different interfaces in order to form functional proteins and, ultimately, HIV. Practically, this is not conceivable, and has been explained as enzymes do not have a high sequence specificity, although it has been postulated that a therapeutically applicable inhibitor has to be specific, and should not inhibit human enzymes of this class of substance. 61. Considering these explanations of the head of the chemical department of the scientific laboratories at Bayer's, it becomes obvious that, theoretically, it is not possible to exactly target the postulated HIV protease. Further, it is impossible not to interfere in the cellular processes of integration and disintegration of a variety of proteins. The inhibition of the active protease in AIDS per se makes sense. However, the pharmacological administration of high doses of distinct aromatic substances is a non-physiological measure, connected with serious side effects which excludes their pharmacological use. Indeed, up to date, the pharmacological HIV protease inhibitors prove to be connected with side effects, which demand the absolute necessity of their replacements by phytotherapeutic mixtures. Apart from side effects such as kidney stones, damage of the liver, and increase of diabetic impairment of metabolism, CMV retinitis and hemolytic anemias, these protease inhibitors, after a short-term administration, also demonstrate a loss of effect on the inflammatory process, which was misinterpreted as a result of an acquired resistance by HIV, as well as an incompatibility with many drugs, especially with the ones of the group of cytochrome P450 inhibitors and inductors. 62. Nutritional possibilities in the prevention and treatment of AIDS. Looking at the formula of structure of the synthetic protease inhibitors, it becomes obvious that these are artificially produced aromatic compounds. As we have suggested lately, polyphenols as well as tannins and flavonoids are phytoprotective substances against harmful external influences. As aromatic substances, they cannot be synthesized by the animal organism. The nutritional supply of a variety of phytopolyphenols to the animal organism has the function of operating as a redox buffer and rebalancing oxidative stress conditions with their catabolic alteration of metabolism to the anabolic-catabolic state of equilibrium. 63. Flavonoids and tannins are effective with respect to 1. Inhibition of lipid peroxidation. 2. Scavenging of oxygen radicals. 3. Binding and inactivation of pro-oxidative transition metals such as Fe and Cu. 4. Binding of proteins including attenuation of their enzymatic activity, protease inhibitors. Upon these reductive activities, flavonoids and tannins are oxidized themselves. A well-known example is the reduction of vitamin E by vitamin C or coenzyme Q. These mechanisms are at the beginning of a cascade of recycling. This example demonstrates that the multitude of almost 5,000 different flavonoids and tannins is used to overcome the oxidative state of the X-antioxidative molecules at the end of the cascade of recycling, by transferring it to a variety of native molecules. The stress-induced state of catabolic metabolism in AIDS is in the center of the pathogenesis. The correction of the connected whole-body inflammations caused by oxygen radicals and protease activation is a compelling preventive and therapeutic action which urgently demands the use of phytotherapeutic polyphenol compounds. Possibilities and limits of treatment of hepatitis in anti-HIV positive individuals. A symptomlessness and the stress-induced activation of liver inflammation in healthy individuals are characteristic of parenterally transmitted inoculation hepatitis, hepatitis B and C. The classic example for this occurrence is post-transfusional hepatitis caused by blood and blood products of clinically healthy blood donors. At the occasion of a study, made in the early 50s at the blood transfusion service of the Swiss Red Cross on recipients of lyophilized pools of mixed plasma of 50-70 healthy blood donors, it was observed that this caused serious, sometimes even lethal hepatitis in many ill recipients, 64 to 66. It is emphasized that in a contaminated organism with parenterally transmitted hepatitis inductors, now called hepatitis B and C, the aim of treatment has to be reduced to just reach a normal state of health. The administration of virucide, cytotoxic drugs is not able to eliminate these inductors from the organism. Proceeding from this knowledge, in Poland, for 2 decades Brosko et al. Have been collecting data with the Tibetan prescription of phytotherapeutic formula, Padma 2867. They showed that this phenol-rich plant compound is able not only to reduce the serum level of hepatitis B antigens in hepatitis B patients, but also to augment the serum level of hepatitis B antibodies. At the same time, an amelioration was observed in these patients regarding their clinical condition and the biochemical and histological results from their hepatitis. Based on these pioneer results, today in patients suffering from chronically active hepatitis, the substitution with phytopolyphenolic mixtures has priority over other treatments. How does the nucleoside analogs treatment of AIDS patients influence their course of disease? After having examined 8 reports on HIV-positive long-term non-progressors, who stayed clinically symptomless for over 10 years, we realized that, without exception, they had not been treated with nucleoside analogs, 68 to 75. We consider this as a confirmation of our above-mentioned caution as to the prophylactic and therapeutic administration of these cell toxics, originally developed for treating cancer, in the autoimmune course of disease in AIDS. Nutritional supply of polyphenolic mixtures as basic treatment of anti-HIV positive individuals and AIDS patients. As initially showed, a positive anti-HIV test is an indication of an augmented formation of autoantibodies against cytoskeletal proteins, i.e. actin. This condition is pathognomonic for chronically active hepatitis. AIDS as serious immunodeficiency syndrome is the expression of a persistent hypercatabolic state of metabolism along with a stress-induced whole-body inflammation. A successful treatment of such conditions consists of the nutritional supply of a sufficient quantity of antioxidative and antiproteolytic phytophenolic mixtures, consisting of flavonoids and tannins. As neither the animal nor the human body are able to synthesize aromatic compounds, they are fully dependent on a sufficient supply of anabolic effective phytopolyphenolic mixtures, in order to adjust catabolic states of metabolism. These mixtures are present in drugs made of teas and spices. Padma 28 proved to be the most effective one. Additionally, it is recommended to balance other possible states of deficiency of vital nutritional components such as polyanions and essential fatty acids. Completing this review, we came across the publication by Padian et al., which remarkably emphasizes the insignificance of heterosexual intercourse in transmitting HIV. In this study, extended to 10 years, the authors say, male-to-female transmission was approximately 8 times more efficient than female-to-male transmission, and male-to-female per-contact infectivity was estimated to be 0.0009. Obviously, AIDS is not a viral venereal disease, but an inflammatory autoimmune process.